So first, you have to tell me if you can hear me. Okay. And even those in the back of the room? Great. Thank you so much for your generous introduction, Christoph. Now I can proudly add myself to the list of people who have had the honor of being introduced by you. Unique introductions. I'm not being facetious. Thank you for that. And thank you for the great openness and warmth with which all of you have welcomed me as a Carson Fellow at the Rachel Carson Center. I'm thrilled and honored to be here as a Carson Fellow. So, having agreed to present at such an early point of my tenure as a Carson Fellow, my focus today will be on the historically grounded research upon which my current project here is based. My talk is going to be about the social and historical dimensions of prevalent understandings regarding colony collapse disorder, or CCD, which is a phenomenon of accelerated and mass honeybee die-offs in the United States. It's a collaboration with Daniel Lee Kleinman, who is a professor of science and technology studies at the University of Wisconsin-Madison. And this work that I'm presenting today is or was funded by the National Science Foundation's Science and Society program. So many of you probably know this. Honeybees are hugely important ecologically and economically, not only for the honey that we have the luxury of tasting, but more importantly for pollination. In the United States, honeybees are responsible for pollinating over 90 different fruits, vegetables, fiber crops. And a world without bees, without insect pollinators, could look something like this in the southern part of China, in a southern province where growers are being forced to hand pollinate in the absence of insect pollinators. And believe me, it's a really arduous task to be pollinating, each blossom. What a beehive could do in very quickly. It takes a whole group of people several days to do. So the dominant research orientation toward understanding colony collapse disorder, or CCD, has been biological, a frame of analysis which takes honeybees as its primary object. While this is important, it's not sufficient because most bees in the United States are managed by beekeepers. They are not narrowly wild. And so bee development, bee growth, physiology, pollination, honey production reflect historically shaped practices in beekeeping, bee biology, and agriculture. Hence my title, Being Human, which implicates human ways of being in honeybee ways of being. And along this revised frame of analysis, which takes human honeybee relationships as its object, Kleinman and I have examined disputes over the involvement of certain insecticides in CCD in order to understand the circumstances under which different stakeholders, different actors are listened to or ignored by established experts. So my talk will proceed as follows. I'll first lay out what the controversy over insecticides is about in the United States. I'll then move on to highlight my findings about the different ways in which beekeepers and bee biologists have approached CCD and the issues that this raises regarding the social organization of knowledge and knowledge production. I'll conclude by highlighting my latest work that this research has provoked. Since 2004-05, commercial beekeepers who rely primarily on beekeeping for their livelihoods have been losing between 30 to 90% of their beehives, a level that's significantly higher than the prior loss averages of 15 to 18%. In fact, last year, 2012-2013, this year has been one of the worst on record in the United States. The widespread consensus among bee scientists is that CCD is not caused by any single factor, but is the result of a complex combination of multiple factors, such as we have pathogens, various kinds, viruses, bacteria, a blood-sucking varroa mite that's seen as a scourge by beekeepers. We have farmer practices like monocultures. We have farmer-applied pesticides, insecticides. We have beekeeper-applied pesticides to kill those mites. So a heady cocktail of factors. The problem has been which of these factors, which sets of factors are more prominent, and how might they be interacting to provoke the losses? That's still unresolved. In particular, there's a major controversy that's been going on about a newer class of systemic insecticides, which persist in treated crops for much longer durations than older pesticides. Some examples that I'll be using in today's talk are imidacloprid, clothianidin, etc. Several beekeepers have consistently noted a correlation between the occurrence of colony collapse disorder in their beehives and the proximity of their beehives to crops that were treated with these newer systemic chemicals. And these beekeepers suggest that the systemic insecticides are a crucial part of the cocktail of factors responsible for CCD, and hence they've been calling, along with environmentalists, for precautionary limitations in the usage of these chemicals. Several scientists, on the other hand, reject the beekeepers' knowledge by citing the lack of conclusive evidence from scores of field experiments by university and industry toxicologists. So regulators in the US Environmental Protection Agency, or the EPA, which is the main governmental agency in the United States dealing with pesticide regulation, the EPA regulators privilege this toxicological evidence from field studies to justify the chemicals' continued usage. My analysis of this debate illuminates how historically shaped differences in the social contexts in which beekeepers and bee scientists are practicing lead them to approach CCD in divergent ways. Commercial beekeepers' approaches to colony collapse disorder are intertwined with the historical trajectory of US agriculture and the rise of the crop pollination business. So here we see on the slide a bunch of beehives that are getting prepped for pollinating. In the background, these almond orchards in Sacramento, California. Managed crop pollination emerged as a systematic need among growers and as a source of revenue for beekeepers around the mid-20th century. And this emergence of the crop pollination business occurred in the context of the following factors. For one, farmers, growers were finding less and less of those wild pollinators that used to give free pollination services. They were not around as much. So for example, here we have Grixty et al. Showing a significant decrease in the number of bumblebee species, wild bumblebees, in Illinois between 1900 and 1950, kind of early to mid 20th century. So first of all, not as many wild pollinators. And we have to see this also in the context of the rise of chemically dependent monocultures. Okay. So by the early to mid 20th century, we are seeing a rise in the average farm size. Farms were getting bigger, and these also tended to be monocultures. And along with farms getting bigger, we have increasing patterns on this red line of pesticide usage. Okay. In fact, even with the acreage, the green line remaining the same, the amount of pesticide being used on that acreage was increasing. Okay. So, increasing amount of agrochemicals, increasing farm sizes, decreasing wild pollinators. This is the context in which farmers turn to beekeepers for bringing in their beehives, for which they would pay rental prices to the beekeepers. Another important context that we have to keep in mind is also an increase in the acreage of pollinator-dependent cash crops. So here we have almonds as another example from California, where there is, since 1980, in fact, there's been a more than 100% increase in the acreage of this monoculture almond orchards. So US is the biggest producer of almonds in the world, and 80% of the world's almond supply comes from California. It's a huge almond crop, and none of that would happen without honeybees. So, in keeping in mind these factors, managed pollination supplanted honey production as the major driver of the US honeybee industry by around the mid 20th century. And perhaps not coincidentally, there has been a spiraling downward of the number of beehives in the US. In fact, between 1945 to 2005 and continuing on till today, the number of beehives has literally halved since the mid 20th century in the United States. So let me give you some idea of commercial beekeepers' practices for pollination. Commercial beekeepers, of course, are manipulating beehives all the time. Right? But their manipulation of beehives for pollination reflect growers' short-term interests in having strong hives that could pollinate the crop in the relatively short duration of crop bloom. And so then we have a situation where several thousand beehives are being trucked by beekeepers all over the US to different crop settings to pollinate those crops. And these beehives are being prepped in advance with a heady cocktail of chemicals, ranging from high fructose corn syrup to stimulate them, feed them, artificial pollen supplements, antibiotics, mite-killing pesticides. We can just keep going on and on down the list. Not only that, this sad story doesn't end there. We also have beehives being exposed directly to industrial agricultural practices in these crop settings. So we have large-scale monocultures which afford only one single source of pollen and nectar for bees. And these large-scale monocultures are also requiring heavy doses of insecticides, herbicides, fungicides. Furthermore, these large crop settings end up having a lot of beehives crowded together to pollinate that large crop. And it becomes a situation for bee parasites and pathogens to have a party, spread around all these beehives. And CCD emerged in such for crop pollination operations. But commercial beekeepers, we also should keep in mind, have in stake the longer term health of their beehives. They need those bees for subsequent pollination operations. And sick bees make for poor pollinators. So beekeepers with these bottom line concerns reckon with beehives that are in real contexts, that are interacting with multiple environmental factors and are affected by those multiple environmental factors in very complex ways. And so beekeepers' practices of gauging the health of their beehives has been constituted by very informal approaches that are unquestionably practical but are in no way definitive from the standpoint of scientists. I'll give you an example very soon. But I also want to make the point that beekeepers' informal approaches to bee health engender knowledge that errs on the side of false positive conclusions. That's a precautionary orientation, where a substance that might be completely safe is considered harmful based on suggestive evidence. Let me give you an example of the informal approach that I'm talking about. Brood pattern is something that beekeepers use to gauge brood health. Okay. So beekeepers look at the overall pattern of brood development on a beehive comb to not only gauge brood health, but also get an assessment of the queen's reproductive health and the beehive's nutritional status. By contrast, bee scientists who would be interested in gauging brood health would do statistically informed comparisons between individual brood in each of those cells, hence the marks here showing individual brood cells. So brood pattern as an informal measure is encompassing multidimensional aspects of a beehive's health. You have queen's reproductive health, the hive's nutritional status, and the brood. Okay. And it allows for a quick assessment. So this brood patterns were influential in beekeepers' perspectives about the effect of systemic insecticides on their beehives. Okay. But it's not just this informal approach, but also a history of long-term, century-long tensions with farmers over their insecticide usage that is reflected in beekeepers' concerns about these newer systemic insecticides. And more recently, debates in France and the European Union regarding the very same set of chemicals. Here we have a picture taken from 2000 France, where beekeepers were protesting outside the French Ministry of Agriculture, talking about problems that they were seeing in their bees because of purportedly the same chemicals. So 8 or 9 years down the line, US beekeepers' concerns are reflecting these prior concerns from France as well. Let me switch to talking about academic scientist approaches now. So university bee scientists in North America have looked primarily at field experiments for definitive causal evidence of the role of these newer systemic insecticides in CCD. And the majority of these field studies of beehives exposed chronically to different levels of these chemicals in the field have found little measurable evidence of harm. And this is a representative conclusion here. Exposure to clothianidin seed-treated canola has no long-term impact on bees. And this is one of the figures from the paper by Cutler and Scott Dupre, who used colony weight, honeybee hive weight and yield as measures to say that there is no difference between those hives that were treated with canola, with clothianidin treated canola, and those hives that were exposed to untreated control. And there have been scores and scores of these field studies. The onus in these field studies is on precise experimental control of all potentially confounding environmental variables toward isolating the effects of the individual factor of concern, in this case being the systemic chemical. So there is this focus on controlling all different factors to isolate the effects of one factor. It's a control-oriented approach, which occurs, I argue, at the cost of understanding the multifactorial complexity in which beehives are existing and from which CCD emerged. This is because intending to focus on the direct effects of individual chemicals, these field studies don't test real-life scenarios where the chemical by itself may not be causing CCD, but may be doing so through progressive interactions with a host of other factors and chemicals. A scenario that Beekeeper's analyses point out and also laboratory studies suggest. I would argue that these issues remain undone, these issues of complexity remain undone in contemporary field experiments, partly because of the career structure of academia. These issues are very hard to measure. We are talking about low levels of insecticides over a long period of time, several weeks, several months, in interaction with other host of other chemicals. These are not easy things to measure. There is a high risk for getting inconclusive evidence, and that high risk is of significant concern for a university scientist thinking about their professional advancement, about securing publications, about securing grants, about getting tenure. And so bee scientists have taken the easy route often of, because that's more easily publishable, where either you use high dose insecticide and you can see easy differences, or you only look at the individual chemical effects as opposed to interactions. Related to this career issue, there is the statistical practice also of basing conclusive evidence on 95% certainty levels. These are beehives. They are superorganisms in certain ways because there is a high level of variability between one beehive to another beehive. So there is high intra-beehive variation, and there is a huge amount of environmental variation in which these beehives are situated. Okay? Using such a conservative statistical practice of 95% certainty means that most field studies would tend to conclude that there are no differences between treated and untreated beehives, when in fact there might be. So this is a false negative orientation. You recall I talked about the false positive orientation of commercial beekeepers. This is a false negative orientation in which is reflected an academic professional norm to overlook potentially valuable findings rather than suffer the professional embarrassment of having to withdraw results later that were shown to be incorrect. That would be really professionally damaging for a scientist. And so as a professional norm, there are these conservative measures set up. And my argument is that this could be involved as a factor in scientists not finding a role for these systemic insecticides. These are matters of social history, not nature, is really the main, one of the main punchlines of these findings. So the control-oriented research norms and practices that I just talked to you about can be actually historically traced to the very unfolding of entomology as a disciplinary science of insecticide development in the United States. Late 19th century and early 20th century state entomologists sought to prove their indispensability, their importance for agriculture by framing crop problems in terms of pest insects and by studying the efficacy of assorted modes of chemical control. Their experiments embodied causal approaches that emphasized rapid, easily quantifiable, lethal effects of individual chemicals on target insects. They were interested in finding out what's the best way to kill an insect. And here's an example of a typical experiment from Atkins 1951 where DDT of a certain formulation was applied on insects, and then 2 days later and 7 days later the index that's looked at is of mortality, of death. You're looking at how many insects they could kill. And you do the same for different kinds of insecticides at different doses. Interestingly, the modus operandi of bee scientists who wanted to look at the effects of these chemicals on bees also followed the same concepts, procedures, measures that were originally geared toward maximizing killing of target insects. And in the process, these bee scientists, early 20th century bee scientists, ended up overlooking the complex ecologies in which beehives were situated. And here we're talking about sublethal effects, okay? Not lethal, not rapid, but over a longer period of time in interaction with the ecological factors in which beehives are. And here's an example of another typical paper by a honeybee scientist who's looking at percent mortality of bees following a certain number of days for these different pesticides and insecticides. Indeed, bee scientists went on to conclude that in general, modern pesticides are less hazardous to honeybees, and although the newer pesticides are used in greater quantities over larger areas and over a greater variety of crops, they can usually be used with safety if the facts from studies and precautions are taken into consideration. So my argument is that this convergence in the experimental approaches of early entomologists and honeybee scientists ultimately reflects the influence of the norms of the agricultural organizations for which bee scientists and entomologists worked. So many of these bee scientists were working at the US Department of Agriculture, same as the entomologists. Quite a few of these bee scientists were working at the extension and land-grant universities in the US. And so their professional stakes were in the agricultural research communities, and that's where these honeybee scientists sought professional recognition. They sought to publish in the Journal of Economic Entomology, which was the premier and still is one of the premier journals in entomology. Now, a historical shift to a sound science approach to regulatory assessments of environmental harm means that the search for definitive causal evidence has also become the yardstick for governmental regulation of pesticides and insecticides in the United States. So with the EPA's concurrence, this control-oriented approach that has come to be accepted by university bee scientists is reinforced. And this convergence in the orientations of academic toxicologists and EPA has served well the companies that make the insecticides. And maybe this is an answer for Christoph's question about why I'm here. This is Bayer Crop Science, and Bayer is in Germany. and Bayer makes some of these insecticides such as imidacloprid and clothianidin. And Bayer, as well as other such pesticide manufacturers, have every reason to support the sort of control-oriented approach that has become established in academia. And none of these actors would deny the possibility of complex interactions involving imidacloprid or clothianidin. The point is that this historically established approach makes it extremely challenging to find acceptable evidence of complex interactions and sublethal effects of harm. And what's more, this approach also justifies dismissing the knowledge and indeed the expertise of commercial beekeepers. So in conclusion, I've argued today that the worlds of scientists and beekeeper research about CCD have been largely separate, with beekeepers' field knowledge pointing to a prominent role for a newer set of systemic insecticides, and scientists' field experiments pointing to a lack of conclusive evidence in support. There is nothing objectively superior about the scientists' field experiments, is part of my argument. In fact, it's not the objective superiority that makes these experiments the established way to proceed, but historical convergence in the professional academic stakes and interests of bee toxicologists and the norms of EPA's regulatory culture. This historical convergence is what is shaping the circumstances under which agrochemical industry actors are able to advance their perspectives over and above those of beekeepers by drawing on largely unquestioned institutionalized approaches. And finally, which understandings are given priority affects both what is known and what is unknown about the putative links between these pesticides and honeybee health. So moving forward, we need a deeper sense of the interactive historical development of beekeeping and bee biology, along with agriculture, in order to understand the circumstances that have led to the current predicament of CCD. And this is what I'm delving into currently at the Rachel Carson Center, with the first phase of my historical project focusing on the development of beekeeping as a specialized profession, as a specialized industry from around the mid-19th century onward. And in a separate new project that just got funded by the National Science Foundation, Kleinman and I will be developing a transdisciplinary deliberative model to enhance the influence of commercial beekeepers, growers, and ecologists in efforts to understand and resolve the ongoing honeybee declines. Thank you.